These are notes on osmosis, diffusion, and active transport. So within a biology context, these three processes all refer to the movement of atoms and molecules into and out of cells. When you take chemistry and physics later in high school, you're going to see some of these terms again, and it's going to be the same process. It just won't necessarily apply specifically to cells. But in biology, that's really the context in which we see these processes. So I want to give you a few vocabulary reminders. You should have had these from physical science last year, but if you didn't or you don't remember, that's okay. But these are some words we're going to be using. One thing I would say is these are a lot of new terms, so if vocabulary is something you struggle with, I would make note cards and quiz myself every day so that you know exactly what I'm talking about or someone else in class is talking about. So the first word is concentration. Concentration is the amount of substance per unit space. And you've seen this in density, for example, amount of grams per milliliter of substance. Parts per million we saw with the concentration of carbon dioxide in the air, so how much carbon dioxide per 1 million molecules of air. In chemistry, you'll see moles per liter. So mole is an amount of molecules, liter is obviously a volume, etc. We talked about it. I was on a walk yesterday with my daughter, and she had a mocha, and all of the chocolate sauce had kind of fallen to the bottom, so the bottom got really too sweet for her, and I said, oh, you know what, it's probably too concentrated, because normally she drinks ice mochas, and so then the ice dilutes the chocolate a little. So dilute is the opposite of concentrated. And so here we have two examples. So in this example here, you actually have the same number of particles of stuff, these yellow particles, whatever they are. If you count them up, there's the same number in this beaker as in this beaker. But in this beaker, there's a much larger total volume. So if you look at the amount of substance per unit of space, you have less. And one way you can sort of say that is, if these two volumes were the same, so this volume here, okay, is about that big, and now let's try to draw an equivalent volume here, okay, more or less, okay, you see with the same amount of space, there are less particles, yellow particles. So this beaker here is less concentrated than this beaker here. On the opposite side, here we have the same volume, but here we have more particles of stuff per unit space, because it's the same volume, so we don't need to draw any boxes. And here we can clearly see there are less red particles. So that's a lower concentration. So you can either have a lower concentration by having fewer particles of substance, or you can have a lower concentration by having a larger amount of space. Okay, so that's concentration there. So then we're going to talk about solutions. A solution is when substances are so finely mixed you can't tell them apart. So often we talk about like salt water. If you pick up some ocean water, you can't tell the salt from the water because it's so finely mixed in. Air is another solution. You can't look at air and tell the oxygen from the nitrogen because it's so finely mixed. A solvent then is a part of a solution. The solvent is the substance you have the most of. So in salt water, your solvent is the water. In air, your solvent is the nitrogen. There's only one solvent per solution. A solute then is everything else in that solution. So in the salt water, your solvent is the water, your solution is the salt. In air, your solvent is the nitrogen, your solute is oxygen and argon and carbon dioxide and probably some water, helium, anything else that's in that air that's not nitrogen. So we're going to be using these terms a lot. So again, if terms are something you struggle with and you don't remember or know these, I would make note cards. Alright, so on to the processes. We're going to start with diffusion. Diffusion is the movement of solute from high concentration to low concentration, or we would say down the concentration gradient. So a gradient, think of a gradient like a hill. So high concentration is the top of the hill, low concentration is the bottom of the hill. So we're going to move down that hill. And this is a very natural, spontaneous process, right? If you put a ball at the top of a hill, it rolls down a physical hill. If you have a high concentration of particles somewhere, they're naturally going to move out to low concentration. And we're going to do some activities in class to show why that happens. Right now I'm just introducing concepts. So in class we did a demo, right, where we put some food dye into a beaker of water. We didn't stir it, we didn't mix it, but we did see that over time those particles just spread out. So we had a high concentration of food dye, and then over time, right, this is time lapse, over time those particles spread out to a lower concentration. And what's happening on the atomic molecular level, so that's our macroscopic level, on the atomic molecular level is you have these particles of dye, and they're all bouncing around. Some bounce out, some actually bounce back in, so they're going in different directions. But overall, the net flow is that those particles of dye eventually bounce away from each other, and so you get them spread out. And once they are perfectly mixed throughout the solution, we call that being in equilibrium. And we find that whatever kind of equilibrium we're talking about, whether it's forces, whether it's temperature, things tend towards equilibrium. Nature kind of wants everything to be nice and equal. So that's diffusion, and we see that some examples are, other than the food dye, is, if somebody's eating an orange in class, which you shouldn't be doing because it's a science class, but eventually you smell those orange particles, that means the particles have actually diffused throughout the air over to where you are sitting. We see this too in biology with carbon dioxide and oxygen moving into and out of cells. If there's a higher concentration outside, then they're going to diffuse into the cell and vice versa. Again, we're going to get into the why of how this happens a little later, but those are some examples of where you see diffusion. Then we have active transport. So active transport is moving particles against or up the concentration gradient. So what we just saw, and it made sense, is that particles should naturally flow if this is a semi-permeable membrane, so meaning a barrier that keeps some things out but lets other things through. A lot of you, when you did your organelle project, researched that the cell membrane is a semi-permeable membrane, lets some things in, keeps other things out. That naturally these particles should eventually kind of flow in this direction, because this is the higher concentration side and this is the lower concentration side, right? That's what would naturally, or we would say in science, spontaneously happen. However, there are going to be times when, for whatever is going on in your body, and the moving of ions, charged particles, is a time when this happens, where you don't want it to be equal. You actually want a difference on one side as compared to the other. And so you want to move particles from this direction over to this side. Now that obviously is not going to happen spontaneously. If diffusion happens spontaneously, then moving particles the opposite of diffusion is going to take some effort, some energy. In biology, where does that energy come from? That energy comes from ATP. So one of the many uses of ATP is simply moving particles from one place to another when they wouldn't normally move that way. And again, we're going to talk about the mechanism by which this happens a little later. But understand that ATP, what we use ATP for, is more than just like moving our arms around and generating body heat. It's controlling what's going on a cellular level. And then we get to osmosis. Osmosis is the movement of water through semi-permeable membranes from a more dilute solution to a more concentrated one. So in a sense, it's the evening out of water. And it has to do with this concept called osmotic pressure that you don't need to get too much into. But let me give you some examples. So if I have this setup here, where I have a much less concentrated solution here and a more concentrated one there, with osmosis, it's actually the water that's going to move to even out these concentrations. So here, I have a concentrated solution, and that's going to be high osmotic pressure because there's a lot of stuff in that water. Here I have a dilute solution, the opposite of concentrated, and I've got a low osmotic pressure. So the water is going to move from more dilute to more concentrated. So I get this system here, where now I have the same concentration, because water's moved in this direction, and equal osmotic pressure. So that's what happens specifically with the movement of water through semi-permeable membranes. Now, in chemistry, you're going to look at osmosis with different fluids as well, but in biology, when we're talking about osmosis, we're only talking about the fluid water. So I want to look a little more detailed at osmosis and cells. Okay, we have three types of solution: a hypotonic solution, an isotonic solution, and a hypertonic solution. And I'm going to give you some examples of what happens with both animal cells and plant cells. So a hypotonic solution, hypo means less than, means that the solution has a lower or less than concentration of solutes than the cell does. So cells have all sorts of dissolved stuff in them. So if we take a cell and we put it in distilled water, for example, then that solution is going to have a lower concentration of solutes than the cell. So what we see happen is water flows into the cell, and it's actually a net flow. Water is also going to flow out of the cell, but less water will flow out of the cell than in. And again, we'll kind of talk about why later. But more water will flow in because it's going from more dilute to more concentrated, the water is. And that cell, if it's an animal cell, can actually burst and break open. With a plant cell, because there's a cell wall, no cell, instead of just a cell membrane, you actually get what's called a turgid cell, and that's normal for a plant. So this is when your plant is nice and healthy and the leaves are standing upright. The reason the cell doesn't lyse is because of that cell wall. So that's providing that structure to keep that cell from bursting when it's nice and beautifully full of water. Now on the flip side, we have a hypertonic solution. So hyper means more than, right? If you're feeling hyper, you have more than enough energy. So that's when the solution has a higher concentration of solutes than the cell. So for example, a cell in very, very salty water, which is why you don't want to drink a lot of salt water if you're feeling really thirsty, right? I'm sure you've all heard the important survival tip: if you're stranded in the middle of the ocean on a rowboat and you have no water, don't drink the water around you, because you'll dehydrate and die. I don't know what you are supposed to drink. Maybe you're supposed to, like, I don't know, kill your rowboat mate and I don't know. Now we're getting gross. Never mind. Okay, so in this case, the water is going to have a net movement of out. Now again, there's going to be a little movement in, but notice I'm making these arrows much smaller than the out arrows because less water will move in than out. And so you have a net flow of water out. Again, if the cell is more dilute than the surrounding, then you're going to have a net flow of water out of the cell. And you're going to get, in animal cells, a shriveled cell. That's what we saw with the corn syrup. And in plant cells, it's called plasmolysed. I don't care if you actually remember these terms. So the lysed and the turgid and the shriveled and the plasmolysed, these are not terms you need to memorize. So then we have, in the middle, we have an isotonic solution. So an isotonic solution is a solution that has the same concentration of solutes as the cell. So iso meaning same. An isobar on a map is a line that's drawn for the same elevation. An isosceles triangle has something about it that's the same. Although my geometry is not wonderful, so I don't know what about it is the same. You should know. So it's a solution that has the same concentrations of solutes as the cell. So a cell in a similarly concentrated solution. And in that case, now in this image, they do have arrows flowing both ways. Water is going to flow both ways, but in equal amounts. So that cell is going to stay the same volume as it was before. And that for animal cells is a normal cell. Now for plant cells, that's actually a flaccid cell. So I am really bad at taking care of my plants, and so every now and then I'll look over at this plant I have on my bookcase and I'm like, oh, the leaves are really droopy. Clearly it needs water. That's what's happened, is these cells are not nice and turgid anymore. They've lost water and they physically cannot sustain themselves. They don't have that matter in them to make them nice and happy. And so I water it, and then the cells uptake the water, because if you think now, the more dilute solution is outside the plant, so they're going to take up water, goes into the cells, and nice, happy plants again. So then one more thing I want to talk to you about, because we see it in society, is using this concept of osmosis to make fresh water. But what it is it's called reverse osmosis, so it's going in the other direction. So normally what we would expect is water to flow in this direction overall. We would expect water to flow from pure water into salty water, right? And that's because water normally would flow from more dilute to more concentrated. However, we have a water shortage in this world. It's not a shortage of water. We have plenty of water. It's just water we can't drink. We just talked about, rowboat. Don't drink the salty water. But the idea is, if we could get pure water from salt water, then we could get more pure water to help the water shortage. And so what they do, and this is an actual procedure, it's used, there's reverse osmosis plants in Australia, in Florida, in Israel. So all around the world, countries use reverse osmosis to get pure drinking water. And so what they do is, similar to active transport where you're using ATP, here we're just applying pressure, and we are literally forcing water through a semi-permeable membrane. Now this semi-permeable membrane allows water to pass, but not dissolved salts. And so you are physically applying pressure to get water to move through a membrane against the normal direction of flow. And so you get pure water. This does have some downsides though. The first thing is, it takes an incredible amount of energy. You are forcing water in a direction it wouldn't normally go. So it takes energy to make this water, and energy is of course an issue as well. The other thing it does is it leaves behind this salty brine. So you end up with a very, very concentrated salt solution. The more water you force through, the more energy you need to force water through. So essentially, the more salty this becomes, the harder it is to get the water through. So at some point, you have this salty brine. So then what do you do with it? If you put that back into the ocean, it changes the ecosystem of that ocean. It makes it saltier. And as you know, back to ecology, different marine animals are adapted to certain amounts of salt. So then you're changing the ecosystem, and you're killing off organisms that depend on that environment. The other thing though is, as you make the water around you saltier by dumping this brine back in, then when you go to collect more water, it's saltier, and now you need to apply more pressure, which takes more energy. So there are trade-offs here, right? The great benefit is you get pure water for drinking. However, the cons of this is it takes a lot of energy, and it takes more and more energy the more and more you do it, and you end up with this concentrated brine that can pollute your environment. And so, going back to there are no perfect solutions, how are you going to get drinking water for a bunch of people who need it, and what are the trade-offs going to be? All right, so those are the processes that we see. There are other processes as well for getting things into and out of cells. As you're doing research, some of you may come across something called endocytosis. We're not going to worry so much about every single process, but these are the three main ones that we're going to focus on: osmosis, diffusion, and active transport.